Hello friends, good evening. Welcome to Sam Biology class. Today we shall discuss about the principles and applications of high performance liquid chromatography. Before that, I request all of you, my friends, please subscribe my YouTube channel. Also watch, like and share my videos with your friends. Usually, a conventional chromatographic experiment takes unusually long time. To reduce the time of experiment, one can increase the flow rate of the mobile phase, thereby reducing the retention time of the sample components. Although this step reduces the time of experiment, it also reduces the efficiency of the column. This is because the sample components undergo lesser number of equilibrations between the stationary phase and the mobile phase. To increase the flow rate, one has to apply a high pressure. Conventional supporting matrix can tolerate a pressure up to a certain limit. At very high pressure, the matrix particles are badly affected, thereby giving rise to flow rate anomalies. All these problems are overcome in high performance liquid chromatography, which is a powerful technique that uses extremely high pressure up to 8000 psi to increase the flow rate of the mobile phase, thereby reducing the time of the experiment. At the same time, it also improves the efficiency of the column. There is no loss in efficiency because the support particles are very small and more or less uniform in size. The lateral diffusion is also less because of two factors. One is the smaller particle size and the other is the enormous pressure. Because of this, the time of experiment is reduced considerably. Because this technique applies high pressure to increase the flow rate, it is also called high pressure liquid chromatography. Although high performance liquid chromatography is primarily used for analytical purpose, it can also be used as a preparative technique. This figure illustrates the major components of a Hplc system. One component is the solvent reservoir which stores the mobile phase. It possesses a solvent microfilter. The second component is a high pressure pump which forces the mobile phase through the column. The third component is the sample injection port which is used to inject the sample into the mobile phase. The next component is the column where separation takes place. The columns that are used in Hplc are made up of stainless steel, glass, copper, aluminium or PTFE. The length of the column is from 25 to 50 centimeter and its internal diameter is from 1 to 4 millimeter. The column should possess an internal mirror finish in order to allow efficient packing. The packing material is supported by a porous stainless steel or a Teflon plug or a disc that is placed at the bottom of the column. The next component is the detector. Because the amount of sample injected into the analytical column is small, it is imperative that the sensitivity of the detector is sufficiently high and stable. Two types of detectors are commonly used in Hplc. One is the UV visible photometer and the other one is UV visible spectrophotometer. The detector reads the concentration of sample components as they come out of the column. The last component is the potentiometric recorder that produces the chromatogram. In addition to this, refractive index monitor and fluorescence detector are also available with the Hplc system. Applications: high performance liquid chromatography is the method of choice for analytical purpose. One of the biggest advantages that it has over other technique is the speed of analysis, which is many times more than other technique except perhaps for gas liquid chromatography. On the top of all this, the detectors are non-destructive. As a result, the separated sample components can be utilized for further study. Hplc has been successfully used for the separation of different biomolecules and other compounds that include proteins, nucleic acids, polysaccharides, plant pigments, amino acids, pesticides, steroids, drugs and metabolites, plant and animal hormones, and also complex lipids. Friends, this is all about the principle and applications of high performance liquid chromatography. For more updates, subscribe my YouTube channel Sam Biology Class.